Hi, and welcome to the Demystifying Research YouTube channel. Today we'll be looking at Enzyme-Linked Immunosorbent Assays, or ELISA. In this video, we'll be looking at what ELISA is, the different components and principles, the different types of ELISA, how to run this assay, the pros and cons, and future directions for this method. ELISA is an incredibly important diagnostic tool used for detecting and measuring soluble substances like peptides, proteins, antibodies, and hormones within a sample. This method relies on a strong, hyperspecific covalent bond between antibodies and antigens to detect their activity within a sample, and has a wide variety of uses in diagnostic, research, and clinical settings. The principle behind ELISA is quite simple. First, you want to identify your analyte, or the molecule you wish to detect. In most cases, this is an antibody, antigen, or another biological marker like a cytokine. Next, you must choose an appropriate capture protein. This is either an antibody or an antigen specific for the analyte you're detecting. So if you're measuring an antibody, you would capture using an antigen or vice versa. The capture protein is immobilized in the wells of the microtiter plate and quite literally captures the analyte for detection. Arguably, the most important part of ELISA is detection. This is done using an enzyme-conjugated antibody that produces a detectable signal upon reaction with a substrate. The conjugated enzyme is most commonly horseradish peroxidase or alkaline phosphatase. However, this may change depending on the biological sample being tested. There are three commonly used types of ELISA, which vary in use depending on your experimental needs. These are direct, indirect, and sandwich ELISA. In direct ELISA, the entire biological sample you are measuring is incubated and all proteins are immobilized onto the microtiter plate. Next, a primary conjugated detection antibody specific for the antigen you are detecting is added, which binds directly to the immobilized antigen. Direct ELISA is quicker than other methods, however, has lower sensitivity compared to the other types. This method is used simply to detect the presence of specific antigens within a sample, for example, testing of hepatitis B surface antigen in blood samples to diagnose Hep B infections. In indirect ELISA, the plate is usually coated with a capture antigen specific for an antibody you wish to detect. Following the addition of the sample and capturing of the antibody, a secondary enzyme-conjugated antibody is added that binds to the analyte antibody. Indirect ELISA offers much greater sensitivity compared to direct ELISA, however, requires a more complex workflow and introduces the potential for cross-reactivity of the secondary detection antibodies. This method is used to detect specific antibodies within a sample, for example, testing for HIV antibodies in the blood and diagnosing HIV infection. Sandwich ELISA differs from direct and indirect ELISA, as rather than using an antigen capture protein, it is an antibody specific for an antigen you wish to quantify. Firstly, the capture antibody is coated onto the plate, followed by addition of the biological sample. The analyte antigen binds specifically to the capture antibody, which can then be detected using a secondary conjugated detection antibody. Sandwich ELISA can be run using a direct or indirect method, depending on how many detection antibodies are used. Sandwich ELISA has the highest sensitivity out of all types of ELISA, however, is the most time-consuming and expensive method. Sandwich is typically used when you wish to measure the concentrations of a specific protein or antigen within a sample, like measuring the amount of insulin within the blood for monitoring of diabetes. Now that we're familiar with the different types of ELISA, let's put it into practice. Say you're a research assistant working in Dr. Manali Mukherjee's lab, investigating different biomarkers for asthma. You wish to test whether concentrations of a specific protein differ between healthy controls and asthmatic patients. What kind of ELISA would you use? If you said sandwich ELISA, you are correct. As the proteins you are detecting are likely present in small quantities, and we wish to accurately quantify the concentrations, sandwich ELISA would be best for our purposes. So let's run it together. First, it's important that the wells of our microtiter plate are coated with our capture antibody, or the antibody specific for the antigen we are measuring. For commonly found proteins, commercially available ELISA kits with pre-coated microtiter plates are usually available. One of the most crucial steps in running an ELISA is preparing your standard curve. This is done by creating serial dilutions to create a range of known concentrations of the analyte you are hoping to measure. This provides a reference when measuring the amount of analyte in the unknown samples. Prior to addition of the sample, we perform a blocking step. The blocking buffer binds to all epitopes within the well besides the epitope of interest. This prevents nonspecific binding of the antigen or detection antibodies. Next, the sample is added to the wells and incubated to promote binding of the antigen to the capture antibody. The plate is then washed to remove any excess and unbound protein. Following washing, the only thing that should remain in the plate is the protein of interest bound to the capture antibody. Now it's time for detection. We can either add a primary conjugated antibody, which binds directly to the analyte, or we can add a primary unconjugated antibody, followed by a secondary enzyme-conjugated antibody, which binds to the Fc region of the primary. Lastly, the substrate is added. This reacts with the conjugated enzyme to produce a detectable signal. And the result is this beautiful color gradient we see here. Depending on the amount of bound enzyme-conjugated antibody, the signal will vary in degree of color produced, with deeper colors representing greater concentrations. The microplate is then read using a spectrophotometer, which measures the light absorbance of each sample at a specific wavelength. The absorbances of the samples can be compared to that of the standard curve to determine concentrations. ELISA is a sensitive technique, meaning it is able to detect the target protein even at very low concentrations. This is quite important in research and diagnosis, where even small amounts of the analyte can indicate disease. In general, ELISA is a relatively quick and simple method, and also allows for high throughput, or a lot of samples to be measured at the same time. Unfortunately, while ELISA is a very promising technique, there are still limitations to its use. The accuracy of this method varies depending on the type of ELISA being used. Being a single-point assay, ELISA can only provide information at a single point in time, making it difficult to monitor changes in analyte levels. Lastly, while prepared commercial ELISA kits are available, they can be costly, ranging from $100 to upwards of $500 per kit. Despite these disadvantages, many efforts are being made to evolve and improve ELISA. Recently, a glass capillary-based microfluidic ELISA device has been developed, which requires a significantly reduced sample volume, much shorter turnaround time for results, and shows a near tenfold increase in dynamic range of detection. Similarly, another recently developed ELISA platform is the ELA device, which also utilizes a microfluidic cartridge design and offers fast, hand-free, reproducible data on one or multiple analytes simultaneously. In conclusion, ELISA is a powerful technique in modern biology and medicine. It allows researchers and clinicians to detect and quantify a variety of analytes in biological samples and can be adapted for many different purposes. Overall, ELISA is a sensitive, reliable, and versatile assay that has revolutionized our ability to understand and manipulate biological systems.